Welcome, everyone. This is the year's first IITE seminar. So, in case you are a repeat offender, welcome back. Otherwise, you're warmly welcome to this club where we nerd out about theoretical ecology because we can and because we want to. So, it is my great pleasure today to welcome Theo Gibbs, who is a postdoc at NYU right now. And he's going to be talking to us about higher order interactions, a topic that has received some attention in the past as well. So, we'll see if there's any synthesis going ahead from there. And without further ado, Theo, please take it away.
Awesome. Well, thank you very much, Yuri. As a repeat offender myself, it's a real honor to give this, presentation. So yeah, I'm excited to get into it. Today, I'm going to talk about whether higher order interactions promote coexistence in diverse ecological communities. So diverse ecological communities are commonplace in nature. Two classic examples are the hundreds of tree species which co-occur in 1 hectare of Amazon rainforest, and then the thousands of bacterial strains which coexist in the human gut microbiome. And both of these diverse communities perform really important ecosystem functions. So more diverse forests store more carbon, and then more diverse gut microbiota better resist pathogens. And so these observations about the importance of diversity for these ecosystem functions have made understanding how the interactions between species maintain diversity one of the really central challenges in quantitative community ecology. So in the traditional approach to this problem, ecologists typically assume that species interact in a pairwise fashion. So I'm going to try to diagram out for you that traditional workflow in this slide. So an ecologist will go out to the field or to their test tube and measure all of the direct pairwise interactions between the species. So here the circles are different species and the red arrows are these direct pairwise effects like competition or facilitation between the species. And then they'll plug these interactions into some ecological model, oftentimes the generalized Lacovotero model, but there are many alternative choices. And then use this model to predict community composition and coexistence. And so in the simulation context, for example, coexistence just means which species have some positive abundance at the end of the simulation. So unfortunately, this traditional approach often fails and instead predicts widespread exclusions, often because of strong interspecific pairwise interactions. So we have a few different lines of evidence for this now. There have been many experiments with annual plants in the field. So in these experiments, the experimentalists will choose some annual plant species that they know to coexist in some set of meadows. But when you measure the interspecific interactions between them, they'll be specifically the interspecific pairwise interactions, they'll be too strong to predict this coexistence. And there's been more recent work with microbes in the lab, and specifically one very nice recent paper from Chang et al. That took a community of 12 different coexisting microbial strains. So they coexist in the lab, but then in the sort of pairwise contest between the strains, the most common outcome was exclusion, pairwise exclusion. So one of the species being excluded by another. So once again, the interactions between the species are very strong, but we still see coexistence at the community level. And, in some sense, we think that this shouldn't happen because of diversity stability theory. So the basic result here is that when the pairwise interspecific interactions between species are very strong or variable, we're unlikely to see stable diverse coexistence. So what explains this mismatch? What explains the mismatch between predictions based on pairwise experiments or pairwise interactions and diverse coexistence? There's many possible explanations, and in ecology, probably the right answer is almost always it depends, or it depends on your study system. But here I'm going to talk about two possible explanations, interaction chains and higher order interactions. And as you can probably guess from the title of my talk, I'm mostly going to focus on higher order interactions today. Okay, so what are these two different possibilities? Well, an interaction chain is some pattern in the direct pairwise effects of every species on every other that allows emergent coexistence, even if the pairwise interactions are strong. So the kind of prototypical example of this is the rock, paper, scissors game, where each species beats every other in a cycle, so that in any pairwise contest, there's an unambiguous winner. But when all three species are grown together, no species can gain enough of an advantage to outcompete all the others. And then there's, of course, more complicated generalizations of these kinds of intransitivities that can apply to systems with many more species than three. By contrast, a higher order interaction occurs when the strength of a pairwise interaction between two species is fundamentally different when you have a third species in the system. So I've tried to draw that here by having this blue arrow, connect to this red pairwise interaction between these two species on the right. So there's, a lot of important differences between these two kind of interaction patterns and interaction phenomena. Maybe the most crucial is that on the left-hand side, these interaction chains are fundamentally pairwise. They can be predicted from pairwise experiments alone. But on the right-hand side, that's just not possible because you wouldn't see a higher order interaction with only two species in your experiment. So, as a result, that means higher order interactions are kind of fundamentally missing from the traditional approach because the traditional approach assumes pairwise interactions. But they're often argued to benefit coexistence. And so one high-profile study which supports this conclusion concentrated on antibiotic competition between three different E. coli strains. So there's one strain that produces an antibiotic that kills a sensitive strain, but there's a third strain in the system which secretes another compound into the environment which can degrade this antibiotic. I mean, it does it to protect itself, but of course, but in this situation, it also protects the sensitive strain, right? And so you wouldn't have guessed this kind of dynamic from doing all pairwise experiments, but it's a very clean example of a higher order interaction. And maybe it's clear here why this sort of higher order interaction could be good for coexistence because you're protecting the sort of weakest strain. But it's not obvious that all higher order interactions should look like that, right? So we can imagine how higher order interactions might emerge amongst some plant species. So here, this green plant is having some competitive impact on this gold plant. But it's possible that when the green plant is competing with this purple plant, its phenotype changes in some way. Its traits or performance shifts. And this could instead potentially amplify its competitive effect on this gold plant. And in this context, it's less clear what sort of implications these kinds of higher order interactions should have for coexistence. And so, as Yuri pointed out at the beginning or in the introduction, these kinds of higher order interactions have been studied for, probably 50 years, maybe longer. But we don't have clear, rigorous theoretical expectations for how these interactions shape species coexistence, especially in diverse communities. And I'd argue, especially in these contexts where they don't have some specific interaction structure motivated by, say, antibiotic competition. And further, we have limited evidence for how they operate in plant communities, even though this is one of the systems where we know the most, where we've measured many times the sort of pairwise effects between a bunch of different species. So these are the two big knowledge gaps I want to try to address in today's talk. So specifically, I'm going to try to answer, I'm going to answer these two questions, which is how higher order interactions affect coexistence in theoretical models, specifically how these kinds of unstructured or generic higher order interactions affect coexistence. And then second, I'm going to ask whether or not higher order interactions structure the dynamics of annual plant communities. Okay. So to answer this first question, I'm going to consider a generalization of the Locke-Volterra model with higher order interactions. I probably don't need to describe this model to this audience, but I still will. So xi is the population abundance of species I, so how many individuals of species I are in the system. And the left-hand side of this equation is the change in species I's abundance over time. So this is a big system of differential equations. And then the right-hand side has three different basic biological processes. Species grow at some intrinsic growth rate Ri, so how fast do they grow exponentially when they're in isolation. Then there's all the pairwise interactions in the system, which are these Aij coefficients. And they just measure how species J affects species I in direct pairwise, in a direct pairwise way. And then, of course, there's some special pairwise interactions, these AII terms, which measure the effect of intraspecific competition or self-regulation, or really just describe the carrying capacities of the species. And then I model higher order interactions in just the simplest possible way by multiplying together triplets of species abundances, xi, xj, xk, so that the coefficient bijk captures the joint effect of species j and species k on species I. Or, alternatively, you can think about the coefficient bijk as being how species k modifies the interaction between species I and j. In this sort of simplest possible formulation, those two, interpretations kind of apply equally well to this functional form. Okay, so this is what the equations look like. The first thing I'm going to do is just set the self-regulation parameters, these AII parameters, to 1, so that exclusions are going to come about from strong interspecific interactions. um, whether they be pairwise or higher order. And then I'm going to take what's now a classic approach in this general field, which is to sample these interaction parameters from probability distributions with specified means and variances. So the idea is to characterize the outcomes of these models in terms of things like the average strength of competition or the variability in competition, as opposed to the specific values of every individual interaction, because they're very difficult to measure. So just to be concrete, I've shown you two different distributions here from which I could sample, say, the pairwise interactions. This distribution on the left-hand side has a relatively small mean interaction strength. So competition in that system, pairwise competition, I should say, is relatively weak. And then the variation in interaction strengths is also relatively small. So the species are in some sense more similar to one another because any given interaction is more likely to be similar to another interaction. And then the right-hand side shows another distribution, which is just the opposite case, where the mean interaction strength is larger, so competition is on average stronger in that system, and then the variation in interaction strength is broader, so the species are, in some sense more dissimilar from one another. So once I set down these means and variances, I can just sample the parameters, the growth rates, the pairwise interactions, and the higher order interactions from those distributions with specified statistics. And importantly, these kinds of simulations or this kind of analytical approach gives us a very formal way to analyze communities without any kinds of interaction patterns or structure. These are really, the only kinds of structure that go into these models are just the average strength of competition or facilitation and the variation around that strength. Okay, so how are we going to determine the effect of these unstructured higher order interactions? Well, the first thing we're going to do is just simulate the model, which of course, we can do. And then wait until these species reach some equilibrium, which it turns out in the parameter regimes I'll show you today that they largely do reach equilibrium. But I'm happy to talk about exceptions or other more complicated dynamics in the question. And then we can just tally up, how many species got, achieved abundance 0.5, say, and make these species abundance distributions, which are kind of a classic plot in ecology. So the histogram says, the bar just tells you how many species of the initial, however many, reached abundance 1 or 0.5, etc. And then I can do this across many different choices of pairwise or higher order interactions and many different interaction statistics. So this is just the same data again, but in addition to these simulations, I'm also going to use the cavity method, which is a technique from statistical physics to predict the species abundance distribution using only the interaction statistics. So of course, the simulations know what every individual interaction was, but the analytical technique throws all that information away and just takes that mean and variance of the distribution. And so this is what the simulation data looks like. And then these are what the cavity method predictions look like. And, I guess the point of the slide is for you to be impressed by them working so that I can not show you simulations for the remainder of the talk or for the most of the remainder of the talk and just show you predictions. But yeah, so you can see they're doing a pretty good job. Okay, so how does it, because it's a bit more of a theoretically minded audience, I wanted to give you a brief explainer of how the cavity method works. I think a good analogy for it is as a kind of invasion analysis, but for diverse communities. So the basic logic is that you assume there's kind of a coexisting state of a bunch of species that coexist together that came about from some assembly dynamics. And then you consider a hypothetical invader to that coexisting state and ask, what is the distribution of abundances that that invader could achieve? And so then you write down a set of equations for basically the mean and the variance of the abundances that that invader could achieve as a function of the statistics of the underlying community. That's the basic idea. And then you make a self-consistent guess, which is that a successful invader is like a typical member of the coexisting state. Because of course, both of these things were hypothetical, right? So now we have a relationship between the typical successful invader and the typical member of the community that lets us write down some self-consistent integral equations that we can solve for the statistics of the whole community. That's the basic idea of the cavity method. There are, of course, some assumptions that this community is sufficiently diverse, and these interactions don't have very much structure, although of course, some amount of structure can be incorporated. And then we make some appeals to the central limit theorem. So we need the statistics of the, the distribution of the interactions to not be pathological for that. But these are, fairly mild assumptions. And then we can just run this machinery and make predictions for the number of coexisting species. So specifically here, because we're predicting the outcome of community assembly, we're really predicting the fraction of coexisting species as our main metric of coexistence. Unlike, say, in Bayes stability theory, where we predict the probability that an equilibrium is stable, here we're going to derive, what fraction of the initial species pool survived to the end of the dynamics. Okay. So we're almost ready to see some predictions from this cavity method approach. But first, I just want to back up for 1 second and ask, what do we expect for pairwise and higher order interactions in very diverse communities? And to do that, I'm going to review some, classic rules for how two species can coexist, the kind of simplest possible case in ecology. So the first rule is that more harmful interspecific interactions between these two species can give rise to fewer coexisting species. And this is oftentimes called the intra versus inter rule in ecology. The idea is just that as the interspecific interactions become strong, they overwhelm the strength of intraspecific regulation, eventually leading to exclusions. And then the second rule is that more variable interspecific interactions give rise to fewer coexisting species. And here the intuition, I think, is fairly simple. It's just that as these, as one of these two interactions, these interspecific interactions becomes much stronger than the other, then one of the species is a much stronger competitor and can exclude the other one. So I'm going to show you that these rules remain useful for communities with, many more species and much more complex kinds of interactions. Okay, so now we're ready to see some predictions from the cavity method. So first I'm going to show you the situation just for pairwise interactions alone. So just the standard Lockable-Terra model. On the x-axis is the variation in interaction strength. So how broad is that distribution from a few slides ago? And the y-axis is the fraction of coexisting species. So out of the initial species pool, what fraction survived? And then the different colors and line types here are different average strengths of competition. Okay, so this is what the predictions from the cavity method look like. And I think there's two basic takeaways from this graph. First is that more harmful interspecific interactions give rise to fewer coexisting species. So that's just a statement that, as I go from red to green to blue, I get fewer coexisting species out. So just like in the two species case. More competitive interactions produce fewer coexisting species. And then the second rule is that more variable interspecific interactions give rise to fewer coexisting species. And you can see that just because all of these curves are decreasing as a function of the variation in interaction strength. So once again, just like the two species case. All right, now I'm going to show you the predictions for higher order interactions alone. And you can see right away that some things are different, but some things are still the same. So once again, all of these curves are decreasing, which means that more variable interspecific interactions are still giving rise to fewer coexisting species. And I think the basic intuition for this result is really just the same as in the two species case. As interactions become more variable, some species are more hurt by competition, some species are less hurt by competition, and then the ones that are less hurt are likely to exclude the ones that are more hurt. And that, basic dynamic plays out if it's, two species interacting in a pairwise way or hundreds of species interacting through pairwise and higher order interactions together. But we found a new behavior for higher order interactions, which is that it's no longer the case that more harmful interspecific interactions always produce fewer coexisting species. That's because there's this crossing over behavior where, the red curve here actually has the fewest coexisting species, even though it's the least competitive system, when the variation in interaction strength is large. And so, in the paper, we did a fair amount of work to figure out why this is going on. So I encourage you to check it out. But, the long story short is that there's a key role for facilitative interactions here that actually reduce coexistence when competition is weak. But, I won't go into it in detail here. Okay, so just backing up for a second, we took this cavity method approach and we found some interesting qualitative differences between pairwise and higher order interactions. But it wasn't like everybody coexisted as soon as we added higher order interactions, even though that was kind of the basic intuition that we might have developed from, say, the antibiotic competition example. At the same time, we know that higher order interactions can generate coexistence because of these, microbial case studies and their associated theory. So I just want to briefly ask, in this same generic model, what kinds of higher order interactions actually do successfully stabilize diversity? So yeah, back to the same, our now favorite model with pairwise and higher order interactions together. And I'm once again going to sample the pairwise interactions from some probability distribution with a specified mean and variance. But now I'm going to choose higher order interactions so they exhibit two new properties. Um, One is that the higher order interactions are going to be forced to produce an equilibrium. So it turns out that a lot of the lack of coexistence we were seeing in the previous part was really due to there being no feasible equilibrium where all the species had some positive abundance. So now I'm going to force the higher order interactions to produce such an equilibrium. And the basic, intuition for what's going on is that if one species is really hurt by pairwise competition, then all the higher order interactions affecting it are going to be facilitative and bring it back up towards, some positive abundance. So that's the first constraint. And the second constraint is going to be that these pairwise and higher order interactions are really correlated at the individual level. So for example, if one pairwise interaction between two species is very strong, then all the higher order interactions that modify that pairwise interaction will also be strong and of opposite magnitude. And now I can just run simulations of that situation and then, plot the results. So here I'm going to show you the probability of stability. So we know there's a feasible equilibrium in this model now, and now I'm just going to plot the likelihood that it's stable. And then these different colors and shapes are just different variations in the pairwise interactions. And then the x-axis here is that correlation strength I just described. So when it's 0, it's like we're back in the previous part and there's no real structure between the pairwise and higher order interactions. It's just the fact that the higher order interactions enforce that there's an equilibrium. But then when it's 1 or when it's large, it means that the pairwise and higher order interactions are really tightly coupled at that individual level. And the big result here is just that as that correlation strength increases, we get diverse systems that are very likely to be stable. And so it suggests that it's possible this kind of correlation strength at the individual level is really an important ingredient for stable, diverse systems produced by higher order interactions. And interestingly, these are all systems that would never have been stable without correlations. Even though, so down here when the correlation strength is small, there's still pairwise and higher order interactions together, but they're essentially never produce stable coexistence. But then when the correlation strength is larger, you do see this kind of diverse, stable diversity. Okay. So just to back up and ask what sort of progress we made on this first question, how do higher order interactions affect coexistence in these theoretical models? We found that unstructured higher order interactions alone did not generate widespread coexistence, but that when we added correlations between the pairwise and higher order interactions, these kinds of interaction patterns could essentially guarantee stable diversity. And this reminded us of these kinds of producer-sensitive degrader examples that I told you about at the very beginning. That's a very clear biological mechanism that could generate these kinds of correlations between pairwise and higher order interactions. So I think one of the big takeaways from these theoretical results is that we really need more experimental measurements of pairwise and higher order interactions together to decide which of these two worlds are we in. Are we in a world where the higher order interactions are unstructured and therefore unlikely to generate lots of coexistence? Or are they very correlated to the pairwise interactions and therefore more likely to stabilize diversity? So I think this is, an important area for the field to go in, but it's also a very difficult one because, inferring higher order interactions is very challenging because of some model inference issues and also just due to feasibility. It's already very challenging to infer all the pairwise interactions in a diverse system. And so modeling and therefore inferring the higher order interactions is just combinatorially worse. So to try to get around those issues, I first wanted to ask just a simpler question, which is whether or not higher order interactions exist, whether or not they exist between annual plants. And so to do so, I designed a model-free test for how higher order interactions, or for whether or not higher interactions operate amongst these annual plants. So here on the picture on the left is, what the actual annual plants look like. And you can see they, they compete in these diverse systems because every one of these toothpicks is a different species. And maybe if you're, a theoretical ecologist, you are attracted to the picture on the right, which is an alternative, description of what these plants look like. And one of the nice features of these annual plants is that they compete in a fairly limited spatial range. So we can measure that empirically. It's like 10 to 15 centimeters. And then by looking, at a picture like this, we can decide which other individuals a given plant competes with. So in this case, this green background individual is competing with four purple individuals and then this, other green individual. And the basic way these experiments often run is that you measure the number of seeds that a given focal individual produces at the end of the season. So that's why I've drawn this, gold individual in the middle and labeled it the focal individual. So in this kind of arrangement, there's just many possible interspecific higher order interactions. So this background green individual is competing with this purple individual, and therefore its phenotype could change in some way, and it could potentially amplify, its competitive effect on the focal individual. Say, this is the same sort of higher order interaction as I told you about at the beginning. And our idea was to try to toggle the frequency or relevance of these higher order interactions by planting these same set of plants in an alternative spatial arrangement. So in this spatial arrangement, this background green individual, for example, just doesn't even know the purple individuals are in the system because of their limited spatial range. So it only sees other green individuals as well as the focal individual. And as a result, we think there's fewer possibilities for interspecific higher order interactions in the arrangement on the left as compared to the right. Of course, there's still, some possible interspecific higher order interactions at the border on the left-hand side, but we think there's many fewer. Importantly, the focal individual sees a very similar competitive landscape in both of these arrangements. And that's because in both cases, they're competing with three individuals of the two background competitor species. So we've really tried to preserve that direct pairwise effect on the focal individuals. And our idea then was to just compare the fecundity of focal individuals to reveal the strength or the operation of these higher order interactions. So let me try to diagram out for you, what the possible outcomes of this kind of experiment could be. So we might measure the number of seeds these focal individuals produce at the end of the season and find that there's no real difference between a clustered background competitor arrangement and a randomly intermixed one. And as a result, we conclude that there's no evidence, for higher order interactions in this case. And I should say, before we ran this experiment, we all thought this was a very real possibility, that these plants would not care whether or not their background competitors were clustered or mixed. And some plant ecologists that we talked to agreed that this is definitely possible. Another possibility is that the focal individuals produce more seeds in the clustered relative to mixed environments. And in this case, we'd have evidence that higher order interactions exist and that they're competitive because the focal plants produce fewer seeds when they operate. And then, of course, we have the alternative possibility, which is that the focal plants produce more seeds in the mixed environments relative to the clustered ones. And so once again, we would conclude that higher order interactions exist and they're facilitative. Okay, so those are the possible outcomes from this experiment. And we didn't just do them with, cartoon dots. We also did them with actual plants outside of Santa Barbara at Cedric Reserve. And I want to shout out Zach Gold here, who co-led this project with me and was absolutely instrumental in its success. Okay, so this is what it looks like with actual plants. Hopefully you can tell the left-hand side is a clustered treatment. The right-hand side is a mixed one. So on the left-hand side here of the clustered plot, we have a bunch of acmospawn individuals all clustered together, and then a bunch of Europappus individuals on the right-hand side. And then through the center line of this plot, we have three different focal individuals of three different species, whose fecundity will measure at the end of the season. And then the right-hand side is a mixed treatment with the same two species, Europappus and acmospawn, all mixed together. And you can't really see the acmospawn here because the Europappus is taller. Okay, so we use 5 different competitor species to produce 10 different background species combinations. And really crucially, we used an equal number and the same identity of competitors between the treatments. So we only ever compared focal plants of the same species competing with the same background species, only the spatial arrangement of those background competitors differed. So we were really trying to preserve those pairwise direct effects between the background species and the focal individuals. And in fact, we went back in and weeded out to prevent any kind of numerical differences between the two treatments. Okay, so we're ready to see some results now. On the x-axis here, I'm plotting the background species combination. So which of the background species which of the species were the background competitors. And in the y-axis, I'm showing you the difference in focal species seed production in clustered relative to mixed environments. So this is some coefficients from a big Bayesian model. And if it's above 0, that means that the focal plants produced more seeds in clustered relative to mixed environments. And then, of course, the opposite case if it's beneath 0. Okay, so those are the results for the first background species combination. And you can see very clear evidence that focal species in this background species combination are producing more seeds in clustered relative to mixed environments, suggesting that a higher order interaction exists between these two background species combinations. So this is exciting. If you've been studying higher order interactions for 5 years of your PhD and you're in your 6th year, it's good news to learn they exist. And in fact, this is the typical outcome across all of the different background species combinations. You can see there's a lot of orange in this graph, and I've colored these densities orange whenever they're above 0. So it suggests that across all these different background species, higher order interactions exist, and they're competitive because the focal plants are producing fewer seeds in intermixed environments relative to clustered ones. There's obviously some variation depending on which of the background species are present. So as an example, whenever acmaspon is one of the background species competitors, or when salvia and Europapus are the two background species, we see a fairly strong higher order interaction. By contrast, when Festuca and Plantago are the background species, it doesn't really matter whether they're clustered or mixed for the eventual effect on focal species seed production. So we like to understand what explains this variation. What explains the variation in higher order interaction strength across these different background competitor combinations? And so we have an idea, which I've sort of been foreshadowing the whole time, which is maybe we can predict the strength of these higher order interactions from changes in the background competitor phenotype. So just like in the introduction, it's possible that these green individuals' phenotype could change in some way when they're competing with a purple individual in the background, relative to a case when they're competing with members of their own species, right? That's our potential mechanistic underpinning of these higher order interactions. And so to try to get at that, we just went into the background competitors and measured a whole suite of performance metrics like height and biomass, as well as more traditional functional traits like specific leaf area and canopy index, which is a metric of how tall up you grow versus how tall out. And then our idea, we didn't know a priori which of these traits was going to be important for higher order interactions. So we just put them all into a big PCA and came up with an aggregate measure of something we call plant phenotype, which is just quantified from this first axis in the PCA. So then we can just ask directly, is a background competitor's plant phenotype very different in one spatial arrangement compared to another? So our question was, could we predict the strength of the higher order interaction across all the background species combinations as a function of how different the background competitors were between the two spatial arrangements? And indeed, we found we could. So here what I'm plotting is the difference in focal species seed production on the y-axis. So just the same sort of Bayesian model coefficients that I showed you, 2 slides ago. And then this x-axis here is the difference in competitor size and traits, as quantified by this aggregate phenotype metric. So when it's 0, the green individuals didn't really care whether, they showed no difference in phenotype if they were clustered or mixed. But if it's large, that means at least one of the background competitors was very different phenotypically in clustered versus mixed environments. And you can see actually a pretty good, correlation here. At least, by ecology standards, this is, pretty reasonably related. So it's exciting. It suggests that there's a potential mechanistic underpinning for how these higher order interactions are coming about. Okay, so I want to briefly summarize the big takeaways from my talk today. We found that unstructured higher order interactions don't generate widespread coexistence using this sort of theoretical approach. Then we found that specific correlations between pairwise and higher order interactions together could essentially stabilize diversity. And then last, we found that higher order interactions are pervasive among annual plants, and they might be generated from this kind of phenotypic shift mechanism. So what progress have we made on this bigger, harder question? Do higher order interactions explain coexistence in diverse ecological communities? We found the theory suggests they can explain it, but only if they have some specific correlation structures. And then we found, experimentally that these kinds of higher order interactions exist, or at least they exist for some systems, which is important, of course. If they're going to explain something, they have to exist. but it's very difficult to test coexistence in this system. So it's a little bit harder to understand whether or not the higher order interactions we found are actually good or bad for coexistence. So I think in the long term, to really try to definitively answer this question, we need to have an experimental system where we can take measurements of pairwise and higher order interactions together, plug them into some more general ecological model, and then use them to predict experimentally observed coexistence and really close the loop from experiment to theory back to experiment. And then we can just do a direct accounting of whether or not higher order interactions explain coexistence in that given system. And I sort of motivated the original, this whole talk by trying to resolve some version of this diversity stability paradox. And I'm really curious to know, from this audience's perspective, what resolutions of the diversity stability paradox can even look like. Because, having worked vaguely in this area for a while, I'm starting to think they mostly look like experimental resolutions. Like you have some experimental system, you make a hypothesis for what explains diversity in your system, and then you test it. But I'd be curious to know from this crowd, if it's possible there can be a purely theoretical resolution of these kinds of diversity stability paradoxes. If we have, say, the existing data and a new theoretical idea that could really make some progress on this deep ecological question. Okay, with that, I just want to say thank you all for listening. Thank you to my collaborators and funding sources. And yeah, thank you for the invitation. I'd be happy to take questions.
Thank you very much, Theo, for the talk. And exactly, I agree. If anyone has a question, please raise your hand in the participant list, and I will call on you as usual. Yes, Michael, please go ahead.
Thank you for the talk. It was super interesting. I have a few questions actually, but I will focus more on the first one. So you said that you found out that higher order interactions, I would say, means more when there is some correlated structure. But my question goes to more to the equation when you have like in the Lutka-Volterra model, like coupling between the two and the three. Did you like to reach this conclusion? Did you examine like all the possible coefficients variations, like if there is some attraction, repulsion? For all the different degrees of freedom and kind of reach the same conclusion? Or it's just for, the interaction that you shown as, for example, or?
Yeah, it's a great question. So in all the theory, what we do is take this sort of agnostic random sampling approach where we say there's an average pairwise strength and a variance in those pairwise strengths, and same for the higher order interactions. And then to sort of build in those relationships, we explicitly introduce like a correlation parameter that constrains those distributions. So I don't want to say, oh, this is, for any more complicated structure of the pairwise and higher order interactions, there, I'm sure, can be lots and lots of different solutions which promote diversity or, erode it, et cetera. But in this random sampling approach, that's kind of what, we found at least that correlations are one good way to stabilize diversity, even though I'm sure there could be others. And part of the motivation for that, I mean, it's very difficult to measure these things, but also when you do measure them, they're pretty heterogeneous. So it's not a terrible way to start, right? I think to say, here's some average strength, here's some variability in those strengths. What does that do? Does that answer your question?
Yeah. I'm just wondering if, I'm just thinking out loud, maybe simulate something similar in, kind of molecular dynamics with many like kind of particles interaction. Maybe it's simpler to kind of, get this simulation and then maybe draw from that another kind of perspective. Just a random thought.
Yeah. So, I mean, I think what you're suggesting is definitely the right thing to do to come up with a more mechanistic picture of how these species are interacting, because I think that gives you a natural way to choose correlations. We were doing this sort of from the top down, asking how would you get coexistence completely agnostic to mechanism? And part of why we're doing that is because, at least in the plant systems, it can be very challenging to nail down a mechanism. But I think that mechanism is sort of the clear next step here. It's, you can only learn so much from these big random sampling approaches.
Okay, thank you very much.
Thank you.
Thank you. Other questions? Yes, Axel.
Hello, Theo. I hope you can hear me.
Yep.
Okay, great. So first on your question, what is this biodiversity stability thing resolved or not? I think it is resolved. We know that the coexistence becomes more difficult with more species based on this cavity method that you use. we know that ecological communities hit the limit that you get at some point where you cannot get more species together. Evidence is all over the world of that this limit is being hit, and so that solves the problem. So the ecological communities self-organize to hit the limit. But I want your opinion on this, but also I want to ask a very biting technical question.
Sure, yeah. Well, maybe so you mean my opinion on the diversity
Yeah.
stability situation? So how do you determine the limit a priori?
It's the transition from the unique fixed point phase to the multiple attractor phase. So
Okay.
there is a singularity where the indirect interactions become self-amplifying, and basically the instability explodes with a certain species.
No, I think I understand. I think I understand the resolution in the lockable Terra model, but I'm curious to know, do you think there's some good experimental systems where okay, this is where we nailed it?
I can send you, I gave a talk about it at some other point. I can send you, there's lots of evidence that exactly this is happening. Okay. Now my biting technical questions. If you show the slide of the Sedgwick reserve, where you have the empirical system and these little dots together, maybe that's the best. So and it's also about indirect interactions.
Yes.
So if I think of a situation that would do first perfect, yes, where the green species suppress the purple species.
Yes.
But the purple species suppresses the focal species, and nothing else happens.
Yes.
It's only direct interactions.
Yes.
Then if the greens are all among themselves, they can't suppress the purple species. Therefore, the purple species will suppress the focal species. While if the green and the purple are mixed, the purple species are suppressed by the green species, and therefore they can't suppress the focal species. Therefore, the focal species benefits from the interaction. It's only indirect interactions, and yet you get exactly the same what you see.
Yes. So yeah, though this is a crucial point that's always good to have a talk. I didn't go into the subtleties, but so what you're saying I think is 100% right. The question is, at what scale do you want to describe the dynamics? So in all of the standard annual plant models, you just describe essentially seed production from year to year, right? And so the kinds of processes you're describing just wouldn't be present in those models. But if you wrote down a model for biomass dynamics across the year, purely pairwise, and then you tally those up in seed year to year, it would be purely pairwise at the biomass level, and it would be higher order at the seed level, which, to be frank, I think is definitely an issue of where you decide to model, right? So, and this I think is a particularly obvious example where the higher interactions, if they come about from that, are not a mystery. It's like the lockable Terra model applied to biomass. I actually think every higher order interaction is like that. I don't think there's any true three-body interactions in ecology. It's a question of where you decide to model. And the kind of ones that you described are maybe less exciting than the antibiotic
on the temporal scale I look at, direct interactions become higher order interactions.
Yes, exactly.
Okay, that's interesting. Yeah. Okay, thank you.
Thank you for your questions.
No, not at the temporal scale. He's saying whether the seeds or the complete biomass. It's not about the temporal scale. It's about how you glob the things together.
I think they're connected because it's
sure. But it was
specifically
to your question. It was whether you're modeling microscopic or macroscopic parts, not time. I mean, time is a whole other can of worms, but I was just correcting that specific.
Okay. Yeah. Thank you.
Thank you very much. I'm very happy that you raised that point because my small forays into higher order interactions, we also made the similar point that it's probably a modeling thing or a modeling choice whether they exist or not on probably very similar grounds. But anyway, Giza, it's your turn. You are muted.
I believe that there are many known. I think is something wrong here with my he?
It's a little echoey, but I can still understand you. Maybe I can change to my AirPods. Is that better? Maybe that's better.
So I'm start again. That I feel very believable the existence of the higher order interactions, but you said that an unstructured something higher order will not have, and I think this is the important point, and even in the diversity stability problem at pairwise interactions, I think the problem is that these models usually assume some unstructured system of interactions. Jury had made some models that even introduction a kind of asymmetry, a kind of tendency for predator-prey interactions in itself increase the stability. So maybe the structure is the point, and the structure is the point in the higher order interactions also, as you said. So the question is that if the structure is the point, then is there a way of characterize that kind of structures which helps coexistence, or it is just can be investigated in a very specific level of interactions? This is, I don't know.
Yeah, I mean, I think I basically agree with you that structure is the key ingredient, and we don't. I guess like part of my ending little salvo was to think, I don't think we have enough experimental or empirical examples with like clear structure or lack of structure. So, for example, like when you go to the annual plant systems, it's not obvious there's a lot of structure in those pairwise interactions, and that could be from it's just too noisy, the data is too noisy, or we didn't collect it at the right, for long enough, etc. On the other hand, like in some of these microbial examples, there's much more structure. And it's sort of clear from the experiments, okay, there's a hierarchy amongst the species, that sort of thing. And so I think that's the big thing that I'm excited to do in the next, however many years I'm still doing ecology, to figure out some of these like pairwise and higher order interaction structures. And I think it would be very like fruitful to do it from some mechanistic picture of, say, like microbial metabolism, competition, cross-feeding, those sorts of interactions, and maybe more detailed than just the classic MacArthur approach, with actual ideas for like genes that are in the bacteria and what they mean for microbial metabolism, et cetera. But I also think it's crucial to do it from real experimental systems where you're going to see if those like sort of hypothesized structures exist. I'm not sure if maybe I'm just agreeing with your question, but yeah.
Thank you. Yeah, Chris?
Hi, yeah, thanks for the talk. Just in terms of the structure of higher order interactions, one of the places where we really do know there is structure is in food webs. So in there, like prey switching and this kind of thing, or resource switching, is one of the few places where we probably can have some sense of guessing how that structure might well pan out. Of course, the problem you described is very hard to experiment on anything more interesting than like a tiny microbe. But what I what I really wanted to ask about actually was something a bit different about your experiment setup in terms of, could you tell whether your higher order interactions that you were measuring were interspecific or just intraspecific? You could almost imagine you could do that same experiment where the purple circles are actually just blanks, and the competition within a species, you get less biomass of that species if they're nearby and clustered, so less pressure, or you get a similar result. Were you able to tease that apart at all?
Yeah, so it's an excellent point that I once again did not describe because of the brevity of the presentation. But it's the kind of thing that we wish yes, it could be all intraspecific higher order interactions. What we're really measuring, which I did not go into detail in, is the difference between the strength of the intraspecific higher order interactions compared to the interspecific higher order interactions, right? That's what we really can isolate. And And so all of our evidence is really at the level of, there is some sort of nonlinear density dependence going on in the background. It could be purely intraspecific. I think we have some evidence that that it's not solely intraspecific because of these background competitor fecundity differences. And then because of, there's a few other layers of evidence in the paper that, I'd be happy to talk about in more detail. But the long story short is that we can't, in a principled way, disentangle those two possibilities. And so the experiment you're describing where you have a blank species was something that we definitely wanted to do, but we just literally ran out of space in the, in the physical plot to do it. I do think that there are some difficulties with that because we know, or at least we hypothesize that there's some sort of nonlinear drop off with respect to just the total number of competitors in the background. And so, we'd have to make those comparisons in a careful way, but yeah.
Yeah. Were you able to make a simple predictor if you just added all the biomass that surrounded the plant, forget the species ID, this kind of stuff? Were any of those sort of indicating that there's something higher coming on, or was it just beyond what was possible to do?
Yeah, so let me tell you what I think the clearest story ecologically is that we found that so we could sort of aggregate across the two spatial treatments and come up with a sort of total competitive metric for how competitively strong was a given background species, whether they were clustered or mixed, right? So some were much stronger than others. And we found that a very clear predictor of the strength of the higher order interaction was the difference in competitive ability of the two background species. So if one is much stronger than the other, we also found a much stronger higher order interaction. Now that doesn't really disentangle these two possibilities, right? It could be that there's a really strong competitor on this side, a really weak competitor on this side, and the focal plant just sort of plastically grows into the weak side and does well. And we didn't quantify that in any way, but anecdotally, we know one of our weakest competitor species was very plastic, so that could be happening. But it could also be exactly the sort of thing that you're saying, that it's just all intraspecific pounding of the background competitor hurting itself, and then that's the big, basically the weak species don't matter, and it's just background competitor interference.
I mean, they are absolutely high order interactions as well. They're just sort of a different, framing of it.
Yeah, exactly. It's more yeah. And honestly, that was something that this really, especially when you put these higher interactions in space, is a lot less clear exactly what we mean by higher interaction, because all of these things would show up as higher interactions in the classic, lockable Terra model, you count up your competition kernel, etc. But maybe they're not really what we mean by higher interaction. So yeah.
Yeah, I mean, I will avoid the terminological dead end that we can go out. Michael's got a question, so I'll...
Before that, may I very quickly interpose, because this was one of the things I was wondering about. So when creating the models, because at least on that level, one can control for this, then all the BIJKs were drawn from distributions. There was no imposing that only I not equal J not equal K not equal I combinations are non-zero.
So in the theory, we try all the possibilities, as we have the flexibility to do. So we do, only interspecific COIs, or we do some with repeated indices as well. Okay. In the cavity theory, the interesting thing is that because the repeated indices are 1/s as frequent as the non-repeated indices, they all sort of go away in the infinite system limit. If you instead have the repeated indices scale with some, so another sort of underlying thing to the cavity theory is we scale all the pairwise interactions 1/s, in the mean and the variance, and then we do another 1/s factor for the higher order interactions. If you were to have the intraspecific or the repeated indice higher order interactions scaled differently, they would be crucially different, right? That would definitely change things.
Okay, but then, sorry, for just that I have it clear, that if I imagine higher order interactions only intraspecific, that would clearly stabilize things. I assume that they're negative. But the cavity method would be none the wiser.
Yeah, let me think carefully about that. Yeah. Would that clearly stabilize things? I mean, if there was variation still, I would expect exclusions.
Oh, well, I'm just make the mean negative enough so that it doesn't matter, even if you moved all.
But if you have if you have I, and then you're saying BIJJ, so
not even just BIII.
Then yes, of course. Yes, of course.
Yeah.
So those definitely are always stabilized.
Yes.
And our results, for example, I think we just got a question like this, like are very sensitive to if you have intra-specific regulation be cubic, and therefore the sort of strength of that regulation, which is not something that we normally think about in ecology, but that definitely changes some of these results. So it's true, I think, what Travis was saying in the questions, like the mean abundance factors in now in a way that it usually doesn't in the lockable terra model, because you have this square going on.
Makes sense. Thank you. And my apologies, Michael, you're up.
Thank you. So I wanted to ask, maybe it's a basic way of thinking about it, but can't you claim that using constraints on the equation of Luca-Volterra, you can induce the power of the higher order interaction? For example, if I say that resources are constrained, the amount of I would say energy kind of in the system. Then if two species are competitive, then the third one should grow because the constraints of the total biomass should be like a constant. Did you try to this is kind of the way you think about structure in that sense?
Yeah, so the kind of thing you're describing is definitely something that like a lot of other ecologists have worked on or tried to do. Have say like a total, have some picture of what the resources they're competing for are, and then if you're competing more strongly between two species, and then this second species competing strongly with the third species, that introduces some constraint on how strongly the first and third will compete, right? Like they'll compete more strongly, say. And those kinds of correlations are definitely not in the theory that I presented, just because it's easier to calculate things about. And for some of the sort of more practical reasons I was describing, where in plant systems we don't exactly know the interaction patterns a priori, and it turns out when you actually just measure these coefficients, they look pretty disordered. But the kind of thing you're suggesting is like something that has been done a lot for the pairwise case, and I think there's like a huge amount of work to do in the higher order case to ask how some of our classic ecological ideas for how species compete constrain higher order interactions. Like that's very much an open field to work in. Yeah.
Okay, thank you.
Thank you.
Thanks.
Other questions? While people.
Oh yeah, sorry, I don't know how to raise my hand on my new Zoom. If there's no other ones, then Travis has another. Sorry. Yeah, so I just wanted to bring up one thing too. People are talking about the strength of interactions, which is fine, but it's really the products of them. So I think we should be cognizant of that, because it's really like the strength of the pairwise. And so you can get really pathological situations where if you have some structure to it, where there's actually no two cycles or higher order cycles, then none of this means anything. It's really about the products of things. And so that's where the pathologies or like the carrying capacity strengths can start multiplying things. And so the scaling really becomes fundamental there about whether you're multiplying things, whether it's the product of a two cycle, right? It's the strength of that. Anyways, I just thought that was important, because it's not just because some people are just talking about like these means of things, and hidden in there though is really this idea of products, right? Feedback cycles, and it's the strength of that feedback that shifts all the eigenvalues, and that's what drives the stability. And yeah, anyways. So for that, I guess the question would be like, did you look at that in your analysis, right? Like how do these strengths of these products, what happens to those? Yeah, did you look at those?
So maybe I don't totally understand what the strength of the products means, but maybe I'll describe a situation where I think I'm doing what you're saying, which is, so in the case where these sorts of more or less competitive, more facilitative, higher order interactions were actually produce fewer coexisting species, we think the basic reason why that happens is that facilitative interactions emerge, which then can sort of drive the some dominant cliques to like basically become very abundant and then suppress all the other species, because within the clique they're facilitating one another, and then outside of the clique they're mostly competing. And that sort of thing really only happens when you have this quadratic scaling of the higher order interactions as compared to the pairwise interactions. It's this kind of nonlinear feedback that really produces those results. And so if we like have the higher order interactions saturate with species densities instead of grow indefinitely with this square, we don't see that behavior. Maybe that's towards what you're arguing?
Oh yeah, no, totally. It's those types of things.
Okay.
Exactly. Yeah. Once again, it's all about the product. So like if you were actually alluding to it before I wrote it, it's You don't see it in the pairwise as much because while pairwise is quote unquote quadratic, you can divide by x, and so it ends up looking almost linear. But really, with the third order term, you then recover this like true nonlinearity. So what ends up happening is interactions truly start scaling by species abundance. So it's like you were saying, if you can get communities to saturate, that means that those interactions then can have more or less strength than they would otherwise. Yeah, and that's precisely what it did. Yeah, you can actually, the shapes of all of these eigenvalue distributions all get determined by the signs of these motifs. Yeah, but yeah, no, that's cool. That's exactly what I was asking about.
Awesome.
Thank you very much. Other questions? While people are thinking, maybe I've thought of something which is maybe it's we're going into speculation territory, but the reason why the examples you gave, for example, with the microbes, with the higher order interactions, the reason why they're so clear and we're so convinced is because they're clearly described via the adaptations of the individual players. So they have clear adaptations, and that makes it clear how they interact with one another, and that makes it super clear why that there is a higher order interaction and how it works. Your empirical study with the annual plants, where you did the PCA, is that a stepping stone towards that kind of understanding, or you, having watched these things in the plots, do you have actually an intuition as a similar kind of adaptive natural history style explanation that would sort of clarify? Do you have such a story or
It's a great question. So I think the annual plants, are very complicated. So I don't want to offer anything as a story that like is of comparable convincingness as the microbes. But I do think that like watching the annual plants, you do see some things that are suggestive. So acmaspon, which was one of the big producers of higher order interaction, and when it's a focal species, is also one of the big deceivers of higher order interactions, is very visibly plastic in its growth structure. So when it grows with itself, it's also the weakest competitor. So when it grows to itself, it grows out like very obviously, and it just does better.
Sorry, by weakest competitor, you mean like an isolation has the highest R star or the less?
It has the weakest competitive effect on the other species as a background competitor.
Okay.
And so when it's growing with itself, it grows like really far out basically, and just kind of clumps up. You can sort of see it in the pictures I was showing. On the left-hand side, it's sort of growing even off of the actual soil onto the tarp. But when it competes with another species, all the other species are taller, and it grows up. So it's sort of like it does obviously worse, and it also grows like directly up instead. So it's now more stiff. It doesn't grow like out onto the ground like this, And so the difficult part about that example is that there's also clearly a performance story there, right? It's doing worse when it's growing up, but it's also plastically changing. So I think the ideal experiment, which is maybe impossible, is to like have some genetic manipulation of some plant where you mess with its plasticity, but ideally control its fitness. That's the hard part. And then you can just do, the sort of classic pairwise experiment trials and see if there's a shift as a function of no plasticity. That would be the ideal. Because I think, these sorts of metabolite stories or these antibiotic stories are very nice. They're very convincing. They're probably not the likeliest higher order interaction across, life. It's probably something generic about plastic responses to competition that was like going to generate lots of the higher order interactions. So they might be dumber than the, antibiotic one, but they're probably much more prevalent. So I think that would be the ideal experiment to do, but it's very challenging to do that.
Thank you.
I could say there's been a few things that have done something a little bit like that using some of the parasitoid aphid plant systems where you can like interact and fiddle around with interaction modifications. But obviously it's a different world to the competition and a setup where you can never get the really big communities. But
Totally. I think in the trophic world, there's a lot of nice examples of sort of behavioral aggregation or behavioral modification as induced by a predator or vice versa that clearly changes your interactions otherwise. Yeah. Thanks, Chris.
Thank you. Any other questions? Yes, Geza.
one short comment. So the adaptation. So I think the structure essentially is the adaptation. Can live together because they adapted to each other, and probably this is the general picture. They adapted either to eat something else or adapted to just eat each other, or there are some arrangements which allow them to coexist. So probably this is the point, central point.
Yeah, I agree. I mean, I think structure could also come from, say, like biochemical networks. Which resources could be produced by what others. If you cleave a molecule, that also introduces structure. But I agree with you that like a clear source of structure will be adaptation and or longer term evolutionary dynamics.
Thank you. Yeah. Any more questions? I can't help but ask, in the experimental setup, how many plots do you need to control for the, I assume, vast variation in seed production and to be able to accurately estimate a good mean with not too much standard error?
Yeah. So it's always better to have more plots. So in that experiment, we did 315 plots, if I remember correctly, 300 and something. And then in each plot, there's 3 different focal plants. So each plot gives us 3 comparisons. But for example, if you wanted to do the like more direct measurement of the higher order interactions where you're trying to fit to fecundity, I mean, one of the nice things about our experiment was there's no density gradient in the background. So we could repeat each treatment something like 15 or 20 times. And that was enough, as opposed to when you're trying to replicate each density. That's very challenging. So yeah, that's part of why I'm trying to work on microbes now.
I very much understand. Okay, sorry. Any more questions? If not, thank you again, Theo. It was a great talk, great chat, and we will be back in 2 weeks. It will be Jonathan Levine giving the next talk, so we hope to see you all there. And until then, have an excellent rest of your days. Bye, everyone.